From time and time again, human ingenuity and talent have made the impossible happen. And this is true in many fields, especially in healthcare. First, by their nature, pluripotent stem cells can potentially be used to create any cell or tissue that the body might need to counter a wide range of diseases, from diabetes to spinal cord injury to childhood leukemia and to heart disease. But before we can do that, all pluripotent stem cells start off as somatic cells. And somatic cells are cells that have already been differentiated, and they all have specific functions already. An example of a somatic cell is a normal skin cell. And iPSCs are somatic cells that manually revert back to their original state of pluripotency. iPSCs serve as a model for scientists in their work to create stem cells from adult cells. It also represents a single source from which all the other cell types from the body can be derived naturally. In order to turn on pluripotent adult stem cells, we need to infect the cells with special laboratory viruses that have been engineered by scientists, professionals, to contain pluripotency genes. iPSCs definitely serve as a model for scientists in their work to create stem cells from adult cells and to do experimentations. Moreover, it also represents a single source from which all the other cell types in the body can be derived naturally. And after the process, cells are reverted back to pluripotent cells and with characteristics of embryonic stem cells. There are huge varieties of applications for iPSCs, but one of the two main ones is iPSCs for macular degeneration. Macular degeneration is a progressive wearing down of photoreceptor cells in the retina leading to blindness. Scientists at the National Institutes of Health Clinical Center in Maryland are testing replacing these cells with healthy retina cells derived from iPSCs. Another great application that scientists are currently taking on is 3D printing organs. By taking a sample of tissues, reprogramming the cells to become pluripotent or iPSCs, and transforming them into cells that make up the heart muscle. Hearts created in that way can completely match the immunological, cellular, biochemical, and anatomical properties of the patient. Patients will not have to wait for a donor, and there will be no rejections of the transplanted organs since they will contain the patient's own cells. As you can see, iPSCs are definitely making a huge breakthrough in the health industry. Thank you.